Hello, my gentle and of course very modern apes. I'm filming kind of up on my desk because lately I've been enjoying filming here when I need to navigate papers and type on my computer, because I normally put my computer when I'm recording back on my set on a little pushy cart, which is hard to type on. For the most part, I'm still gonna record back there, but when I need to get through papers, I'm gonna record here. I also like that you can see the whole shelf. Normally you don't get to see the whole shelf. You don't get to see the wonderful bovid skull that I have up there at the top, or the many hominins that I have here at the bottom that aren't in my primary lineage here. I don't know why I'm telling you all this. Why indeed is the question. Why chromosome is the question. We're talking about the Y chromosome today. So a little bit of background, and I'm going to be as brief as I possibly can be. In conventional science, humans are thought to descend from a more basal ape roughly 7 million years ago. And that's the common ancestor that we share with chimpanzees, the panins, our closest living relatives. So what that means is that if human evolution is true, we should share an immense amount morphologically and genomically with these guys, with chimpanzees and bonobos. This is in contrast to the creationist, or specifically the young Earth creationist proposal, that humans are like these distinct creations set apart from the rest of the animal kingdom, and that the rest of the animal kingdom fits neatly into these created kinds, these Berrimans. You might be new here, so allow me to explain just very briefly. Created kinds, the Berrimans, are this idea, usually within a young Earth creationist context, that in the beginning, roughly 6,000 years ago, God creates these archetypal kinds, and these kinds can beget a lot of diversity that ends up being responsible for all modern diversity and also the diversity that we see in the fossil record, but the kinds themselves are not related to one another. So for instance, God might poof an archetypal felid, a cat, into existence, and that means that all living cats and fossil cats descend from that cat, but none of those cats are related to any dogs, and the dog might be a separate archetypal creation. In this context, right, God creates humans, and humans are one thing, and the rest of the apes are another thing. Or maybe he has a panin kind, like the chimps and the bonobos, and then one for gorillas, and then one for orangutans. And obviously, right off the bat, that's weird, because who would look at chimpanzees, orangutans, gorillas, and suggest that none of them are related? But I digress. This is important for what we're talking about today. Creationists have long been up a creek with regard to the morphologic similarity that humans share with the rest of the apes, right? We have all of the primate synapomorphies, all of the catarrhine synapomorphies, all of the ape or hominoid synapomorphies, and all of the great ape or hominid synapomorphies. That lands humans physically, with regard to our bodies, firmly in the great ape family of Hominidae. And to make matters worse for them, we see the emergence of the unique human characteristics emerge through geologic time in the hominin fossil record. So this is something that's been recognized since Linnaeus, right, the father of all taxonomy and a creationist himself, who well before Darwin recognized that humans and the rest of the apes share a lot in common, more in common than some other groups that he would argue were also patently related, and even reluctantly said, "Yet man does recognize himself as an animal. But I ask you and the whole world for a generic differentia between man and ape, which conforms to the principles of natural history. I certainly know of none. If I were to call man ape or vice versa, I should bring down all the theologians on my head, but perhaps I still should do it according to the rules of science." But with the 21st century came the advent of genetics, and it is here that the creationists placed their hope. Maybe there was something internal about the DNA that separated humans from other apes, something that made them distinct. And then the human and chimpanzee genomes were sequenced. In the early 2000s, we found out that humans and chimpanzees are 98.8% similar when looking at coding base pairs, and around 95 to 96% similar when looking at the entire genome. For the record, that is more similar than lions are to tigers, African elephants are to Asian elephants, and rats are to mice. Some creationists, like one Jeffrey Tompkins, have attempted to, play down this number using their own work, but extensive investigation here on this channel and elsewhere have revealed that Tompkins' methodology fails at just about every level, which is supported by the fact that creationists, for the most part, seem to cite him less and less every year. So we're stuck with this then, the creationists are, that humans and chimpanzees are almost 99% similar in the coding base pairs of the genome, and at least 95 to 96% similar in the entire genome as a whole. And this is a greater similarity than many pairs of animals that creationists would happily insist are related because they are a part of the same kind, like lions and tigers, African and Asian elephants, or rats and mice. But then, in 2013, a new paper came out, one that was quite spinnable. The paper was titled "Chimpanzee and Human Y Chromosomes Are Remarkably Divergent in Structure and Gene Content" by Hughes et al. This was the first real attempt at comparing the Y chromosome between humans and chimps because the Y chromosome is notoriously very difficult to sequence due to tons of highly repetitive sequences. This wasn't the full sequencing, but it was a start, and it found something unexpected. The human and chimp Y chromosomes were far more different than what was expected. The abstract specifically says they found they differ radically in sequence structure and in gene content, implying rapid evolution during the past 6 million years, and they call it an extraordinary divergence. The figures in the paper are striking. They lay out a portion of what they sequenced and compare the human and the chimp, and in fact they are radically different, with the chimpanzee portion also being much smaller, seemingly dwarfed. The authors note how odd this is compared to the rest of the entire genome, specifically displaying the difference between the human and chimpanzee Y chromosome, which are again extraordinarily divergent, and the human and chimpanzee chromosome 21, which are almost identical. They don't mince words, and in the body, they say, indeed, at 6 million years of separation, the difference in the Y chromosomes in chimpanzees and humans is more comparable to the difference in autosomal gene content in chickens and humans at 310 million years of separation. The creationists went ballistic at this, they absolutely loved it, and naturally just skipped the portion in the conclusion that attempts to explain why this disparity might exist, specifically pointing to sperm competition in the pan as opposed to Homo sapiens. This is true. Panins, the chimps and the bonobos, have a drastically different social system and reproductive strategy as compared to Homo sapiens, which probably explains why these differences exist on the Y chromosome and seemingly nowhere else in the genome. This is important as a prediction, and we'll come back to it later. But needless to say, the Y chromosomal dissimilarity between humans and chimpanzees excited creationists greatly, particularly because it was relatively unexpected. Finally, a single part of the genome that was as different as the creationists wanted it to be. Except not actually. The primary difference between the human and chimpanzee Y was actually their size. The chimpanzee Y chromosome had numerous large-scale deletions, and after that it had multiple different inversions and frame shifts and swap arounds that made it incredibly difficult to align to the human counterpart. When you actually looked at the identity of the sequences between the human and chimp Y, they were as similar as expected. Additional work that included a preliminary look at the gorilla Y chromosome showed that gorillas and humans were actually more similar in gene architecture, and humans and chimps were more similar in sequence identity. A geneticist friend of mine illustrated it to me like this. Imagine you have a VHS copy of Harry Potter and the Sorcerer's Stone, and then that VHS copy has two other versions of the movie. Right? In one version, it is exactly the same as the first version, except the last 10 scenes are cut from the film inside the VHS tape. And in the third version, it's exactly the same length as that first copy, but instead of using wands every time the wizards pull out their weapon, they've got like a Glock. They've got like a gun. In what is actually happening within the tape, within the film, 1 and 2 are far more similar than 1 and 3 are, despite the fact that 1 and 3 are the same length. And in genetics, it is the sequence identity and not the size of the sequence that determines how closely related two things are. That's how we do it when we're making pedigrees using sequenced genomes, after all. Add on top of that this is the Y chromosome, right? So this would suggest in kind of a roundabout way that male humans are very divergent from chimps, but female humans are not. And specifically, the gene content difference that did exist between the human Y and the chimp Y, those genes were expressed most commonly in the testes. So the idea that that would be the delineation point for the humankind and the rest of the ape kind is very odd to me. Now, needless to say, creationists didn't care about all of this minutia. They simply liked to point at the Y and say, haha, see, it's too divergent between humans and chimps for them to share a common ancestor. Or, haha, see, isn't it weird that the human Y is more similar to the gorilla Y than the chimp Y? Isn't that suspicious and surprising?
The Y chromosome dissimilarity, this preclusionary line of evidence that suggests evolution simply can't be the case. Because when you consider overall architecture, gene content, size differences, your human Y chromosome and chimpanzee Y chromosome, they're only about 35 to 40% the same. They're half the size, ton of the gene content's just totally different.
I specifically responded to both of those points in videos multiple times early on in this channel. And for a while, they didn't really talk about the Y, at least the YouTube ones didn't talk about the Y, but it seems as though lately they've had a little bit of a resurgence, which is exactly why I've decided to come back and kind of bust this topic. Well, that, and the fact that a new paper came out recently that absolutely beats it to smithereens for me. So when you boil it down, the Y chromosomal argument from creationists can kind of be summarized as point one, there was an unexpected thing in evolutionary biology. The Y chromosome between humans and chimps was significantly more different than we expected, therefore evolution bad. They don't provide a mechanism or any kind of like experimental reason as to why the Y couldn't be that divergent. They just point at it and say, unexpected, that is bad. So point number one should be, we need to see an actual mechanism as to why they propose the Y chromosomes couldn't become that divergent, but we'll get there when we get there. Point two is this sort of inference, this sort of soft implication, I should say, that the Y chromosome might be what separates out the created kinds, at least amongst the apes. Now, 4 years ago, when I covered this topic previously, I pointed to a paper that I felt did a good enough job absolutely quashing this argument. And that was this paper from 2016 by Oetjens et al. Titled Y Chromosomal Structural Diversity in the Bonobo and Chimpanzee Lineages. So this specifically has to do with the structure of the Y chromosome, which again is primarily what is causing this vast divergence between chimpanzees and humans. So 3 years after that first paper came out by Hughes et al., we get this one that looks at the same differences between chimps and bonobos. And this paper is super important for two primary reasons. First, because they tell us how different chimps and bonobos are, at least roughly speaking. We're going to get to the specifics later. But second, they provide some pretty solid support for sperm competition being the reason as to why these Y chromosomes within the apes are so divergent. So the first little bit of this paper that I just want to show you briefly, because we've got a bigger paper that I would prefer to get to, is down here in the results section. It's here, figure 1. Y chromosomal tree and amplicon copy number. The phylogenetic relationship among sequenced pan and human Y chromosomes is illustrated with neighbor joining trees using a Tamura-Nye substitution method. So what we can see here is a comparison of humans, bonobos, eastern chimps, western chimps, and North Cameroon chimps. No, Nigerian Cameroon chimps here. And you can see a massive amount of diversity just within the pannins. So the human Y chromosome is down here, and we see all of these other chimpanzee or pannin Y chromosomes up here, displaying an extreme amount of diversity, with some being like double the size, looking at, I believe, amplicon copy numbers in particular. This is super important because this suggests that there is a massive amount of selection going on within the pannans as compared to some of the other apes that is likely what is responsible for the difference between any given chimp or bonobo and a human on the Y chromosome. This is an explicit evolutionary prediction that was made by Hughes et al. 3 years earlier that is starting to come to fruition here. Because the prediction is very simple, right? If the reason for the difference between humans and chimps on the Y chromosome is due to high amounts of selection, sexual selection in particular, for sperm competition in the pannans, then what we would predict from an evolutionary standpoint is that they should also differ drastically because that sperm competition is going on explicitly in the pannans. It's continuing to act and has been acting since we split with the pannans 7 million years ago. So they should be drastically diverging from one another as well. The creationists, on the other hand, should have no reason to suppose that the Y chromosomes within a kind should be so different from one another. Now, to make something clear, right, this paper wasn't looking at the whole Y chromosome within bonobos and chimpanzees. That hadn't been sequenced yet. They were just comparing amplicon number here, or amplicon copy number. They say our data reveal that amplicons continue to evolve in chimpanzees, leading to distinct gene content found in the Eastern, Western, and Nigerian Cameroon chimpanzee Y chromosomes. This finding represents an important step in our understanding of the evolution of sex chromosomes, specifically in the early stages of primate speciation. Although identifying the specific causes of functional diversity is challenging, we suggest that meiotic drive during spermatogenesis may be increasing DAZ, RBMY, and TSPY diversity among chimpanzees and may be a contributor to their ongoing subspeciation. So that's like the point here, right? Because again, they're pointing to sperm competition as the reason for why these Ys are getting so divergent. They continue here at the end by saying, in contrast, talking about humans here, the analyzed human sample, which represents a 6 million year old divergence, displays uneven copy numbers by position across amplicons and genes. While the relative quantity of the annotated amplicons generated from short read data is reported here, assemblies would provide the spatial diversity of amplicons. With assemblies in hand, one could recreate the non-homologous recombination events that gave rise to the structural diversity within PAN and further clarify the contribution of ampliconic sequences to subspecies evolution. Paper number 1 recognizes that the chimp and human Y are extremely divergent and softly pitches sperm competition is the reason why. 3 years later, we have a look at amplicon copy number variation in chimpanzees, bonobos, and humans, and they provide like a little bit of additional support that sperm competition may be the reason as to why the pannans seem to be evolving such drastically different Y chromosomes from humans and from one another so quickly. But this paper in 2020 titled Dynamic Evolution of Great Ape Y Chromosomes helped elucidate the situation a little bit further. This is a really good paper, and I've mentioned it a couple times on this channel, but I've never really delved into it. In the significance section here, they talk about how the Y is repetitive, which is why it's such a difficult chromosome to sequence, and the evolution of it is difficult to characterize for that reason. They say here we compared the Y chromosome of great apes, human, chimpanzee, bonobo, gorilla, and orangutan, so we've increased that sample size, and found that many of their repetitive sequences and multi-copy genes were likely already present in their common ancestor. Y repeats had increased intrachromosomal contacts, which may facilitate preservation of genes and gene regulatory elements. Chimpanzee and bonobo, experiencing high sperm competition, underwent many DNA changes and gene losses on the Y. Our research is significant for understanding the role of the Y chromosome in the reproduction of non-human great apes, all of which are endangered. So there's an importance here outside of just human evolution and the behavioral ecology of other apes, but also for conservation. So the abstract outlines it quite neatly as well. They give us some background, again, talking about how difficult it is to actually sequence. And then they say the Y chromosomes of great apes, represent a particular puzzle. Their gene content is more similar between human and gorilla than between human and chimpanzee, even though human and chimpanzee share a more recent common ancestor. So we are, again, bringing this point up. This is the one the creationists love. And as always, the abstract points out the problem and then continues to say, To solve this puzzle, we constructed a dataset including Ys from all extant great ape genera. We generated assemblies of bonobo and orangutan Ys from short and long sequencing reads and aligned them with the publicly available human, chimpanzee, and gorilla Y assemblies. Analyzing this dataset, we found that genus Pan, which includes the chimp and the bonobo, experienced accelerated substitution rates. Pan also experienced elevated gene death rates. So now, and we're going to go over this a little bit more in just a second, not only do we have a hypothesis for why the Y is so divergent between hominins and panins, that being sperm competition, but we have a mechanism demonstrated for what creates that difference. So sperm competition is the pressure, accelerated substitution rates, and elevated gene death rates are the actual mechanism by which that Y chromosome changes. Then they say these observations are consistent with high levels of sperm competition in Pan. Furthermore, we inferred that the great ape common ancestor already possessed multi-copy sequences homologous to most human and chimpanzee palindromes. Nonetheless, each species also acquired distinct ampliconic sequences. Well, now wait a second, that's just like that previous paper we looked at, confirming previous work in science. That's crazy. We also detected increased chromatin contacts between and within palindromes, HIC data, likely facilitating gene conversion and structural rearrangement. So now we have a gene conversion and structural differences mechanism. Our results highlight the dynamic mode of Y chromosome evolution and open avenues for studies of male-specific dispersal in endangered great ape species. In the introduction, they give this nice paragraph here that I think nicely lays out the problem that the creationists like to point to, a problem that now has an extremely feasible solution to it. By 2020, they say among great apes, the Y has only been assembled for humans, chimps, and gorillas. A comparative study of these Y assemblies uncovered some unexpected patterns which could not be explained with the data from these three species alone. So remember that bonobo chimp paper was looking only at specific regions of the Y. So at this time, 2020, only the human, chimp, and gorilla had been sequenced. And we didn't quite have a for sure answer. We had a good hypothesis. Despite a recent divergence of these species, that being humans and chimps, that being humans and chimps, sorry, I thought they were going to try to do gorillas too here. Their Y chromosomes differ enormously in gene content and in size, in sharp contrast, that's important for the creationists that they always skip over, in sharp contrast to the stability of the rest of the genome. It's like the fact that they're so ridiculously similar and stable in that way. For example, the chimpanzee Y is only half the size of the human Y, and the percentage of gene family shared by these two chromosomes, 68%, that split 6 million years ago, is similar to that shared by humans and chickens that split 310 million years ago. So a little bit of difference between 6 to 7 and 310. Puzzlingly, in terms of shared genes and overall architecture, the human Y is more similar to the gorilla Y than to the chimpanzee Y, even though the human and chimpanzee share a more recent common ancestor. So obviously from an evolutionary perspective here, the thought would be, the prediction would be, and indeed was, as we previously saw, that something is happening in the Panin lineage, right? The condition was shared by the common ancestor of humans, chimps, and gorillas, and then something changed in the panins, the chimps and bonobos, to the exclusion of the gorillas and the humans that caused them to be different, and that it remained relatively stable in humans and gorillas in comparison. Y chromosomes from additional great ape species should be sequenced to understand whether high interspecific variability in gene content and architecture is characteristic of all great ape Ys. In their methods section here, they talk about what they actually did, which is compare the Y chromosomes of the 5 species from 4 great ape genera. And what's interesting is, like, these are draft assemblies. That's going to be important for later. But even then, they were able to draw some pretty important conclusions. This comparative data set allowed us to answer several pivotal questions about the evolution of great ape Y chromosomes. First, we assessed the lineage-specific substitution rates and identified species experiencing significant rate acceleration. So that's your panins, chimps, and bonobos. Second, we determined the interspecific gene content turnover. And third, we evaluated the conservation of palindromic sequences and examined chromatin interactions with ampliconic regions. Our results highlight the highly dynamic nature of the great ape Y chromosome evolution. So I'm going to make something clear here because that's a lot. The low percent similarity between humans and chimpanzees and their Y chromosomes can be boiled down to differences in size, gene content, and architecture. Size is obvious. The chimpanzee Y chromosome is half the size of the human Y chromosome. Gene content refers to the genes that the nucleotides code for, and those differ between humans and chimps. They're more similar between humans and gorillas than between humans and chimps because explicitly chimpanzees have had high rates of gene death. That kind of goes back to the fact that their Y chromosome is half the size. And lastly, the architecture is different. So the chimpanzee, probably also again due to gene death and high substitution rate, has had a lot of things swapped around. They're not where they should be. This idea of having a greater overall similarity between humans and chimps in sequence identity, but not in gene content, can be a little difficult to understand. So allow me to give you a crude analogy here, a crude example. On your screen, you can see a sequence for humans, chimps, and gorillas. X's represent genes. In gene content, you can see the human and the gorilla are more similar than either is to the chimp because the chimp has experienced 2 gene deletions. However, in overall sequence similarity, the chimpanzee and the human are more similar than either is to the gorilla. As such, humans and chimps can differ in size, gene content, and architecture while being overall more similar to one another in sequence identity. And as for the other differences, this paper quantified and provided mechanisms for all 3 that supports a previous hypothesis. So I want to show you some of those actual sequence alignments, like the numbers that we have, instead of just showing you that nice little example. The example serves to show you what I mean, but let's look at the actual material here. So we have our alignments, and then we have supplementary material, and we're looking at a couple of different things here. We're looking at multi-species and species-to-species alignment. So that's, species A to species B, that's a species to species, whereas multi-species would be like we're comparing the gorilla, the human, the chimp, the bonobo, and the orangutan, like all at the same time. And over here in the supplementary material, we're looking at the proportion that aligns here and here for last Z alignments pairwise, that's your species to species, and multi-species, that's all together. And then the average identity, so the percent similarity more or less for multi-species and pairwise respectively. So what they say here is, as was shown previously, the gorilla Y had the highest percentage of its sequence aligning to the human, which is interesting because when we go over here and we see it, right, it's proportion of what's on the X here to what's on the Y in this table. And indeed, the gorilla is 75.45% in its alignment to the human Y, and then 66 for the chimp, 64 for the bonobo, 61 for the orangutan. But the human over here has the greatest proportion of alignability to the chimp than the bonobo. So the gorilla is more similar to the human, but the human is more similar to the chimp when it comes to what can be aligned. In terms of sequence identity, chimp and bonobo Ys were most similar to one another, 99.1 to 99.2 and 98% for multi-species and pairwise alignments, while the orangutan Y had the lowest identity to the other graded Ys. That is precisely what is expected under evolutionary relationships, right? That all of the African apes, gorillas, chimps, bonobos, and humans should all be more similar to the exclusion of the orangutan, and that 2 species within the same genus, so Panphoniscus and Pantroglodytes, bonobos and chimps, should be more similar than anything else is to one another. For multi-species alignments, the human Y was most similar in sequence to the chimpanzee or bonobo, respectively, less similar to the gorilla, and least similar to the orangutan, in agreement with the accepted phylogeny for these species. So moving back over here, that is precisely what we see. Moving past the proportion aligned to the average identity, the human Y in multi-species is most similar to the chimp, followed by the bonobo, followed by the gorilla. And indeed, this goes for the pairwise alignments as well, being 95.6% similar to the chimp, 95.19 to the bonobo, 95.01 to the gorilla, and 92.46 to the orangutan. So what about this is not directly in line with evolutionary predictions, right? And remember, as far as those differences go, this paper proposes and supports with experiments and data a mechanism. And they propose that the reason why the chimp and bonobo, the pan and Y chromosome, is so different from humans is because of, in part, substitution rates. We next asked whether the chimpanzee Y chromosome, the architecture and gene content of which differ drastically from the human and gorilla Ys, experienced an elevated substitution rate. So here's where they give their substitution rate estimations, and the mechanisms, the methodology for that is all very much above my pay grade, but this is an open access paper, so you can check it out if you want to. And the results are, using the gorillas as an outgroup, we observed a significantly higher number of substitutions that occurred between chimpanzees and gorillas than between humans and gorillas. For autosomes, this number was 0.6% higher in the chimpanzee gorilla than in the human gorilla, whereas for the Y, it was 7.9% higher. Similarly, we observed a higher number of substitutions that occurred between bonobos and gorillas than between humans and gorillas. This number was 2.9% higher in the bonobo gorilla than in the human gorilla, comparison for autosomes, and as much as 9.6% higher for the Y chromosome. Thus, while the pan lineage experienced an elevated substitution rate both at autosomes and the Y, this elevation is particularly strong on the Y. This is only the case if you've got some serious sexual selection going on. And the proposal that they've given, like 7 years before this paper was sperm competition, and that matches precisely. They talk about gene content evolution here. Utilizing sequence assemblies and testes expression data, we evaluated the gene content and rates of gene birth and death on the Y chromosome in 5 great ape species. So we get their methodology here. Again, this is all quite a bit like above my pay grade, right? I don't do genetics very often, and it takes like a lot for me to review these papers and then present them to you in an accurate way. But they do say here, to summarize, the pan genus exhibited the highest death rates for both X-degenerate and ampliconic genes across the great apes. Additionally, we observed significantly higher non-synonymous to synonymous rate ratios for 4 X-degenerate genes and 1 ampliconic gene in bonobo, chimpanzee, and or their common ancestor. However, none of these ratios were significantly greater than 1, providing no evidence for positive selection. So here we have a comparison of like the genes that are proposed in a common ancestor, which genes are getting deleted in different lineages, and which genes are getting added. So this is interesting here. In the human versus the chimpanzee, in the human versus gorilla, we see the addition of the VCY gene, but then that VCY gene is lost in the bonobo. And indeed, we get many genes lost in the common ancestor of the panens, and then some specifically lost in the chimp as well. So again, on those mechanisms to explain the change that we see, the discussion just has some phenomenal quotes. They say higher substitution rates on the Y than on the autosome, which we found across the great ape phylogeny, confirm another study and are consistent with male mutation bias likely caused by a higher number of cell divisions in the male than the female germline. Higher autosomal substitution rates that we detected in pan as opposed to homo lineages corroborate yet another study and can be explained by shorter generation time in pan. A higher Y to autosomal substitution rate, i.e. stronger male mutation bias in pan than in the homo lineage, as observed by us here, could be due to several reasons. First, species with sperm competition produce more sperm and thus undergo a greater number of replication rounds, generating more mutations on the Y and potentially leading to stronger male mutation bias in species without sperm competition. They have a citation there. Consistent with this expectation, chimpanzee and bonobos experience sperm competition and exhibit strong male mutation bias as compared to with no sperm competition, and weak male mutation bias in humans and in gorillas. Contradicting this expectation, orangutans have limited sperm competition but exhibit strong male mutation bias. Second, a shorter spermatogenic cycle can increase the number of replication rounds per time unit and can elevate Y substitution rates, leading to stronger male mutation bias. In agreement with this explanation, the spermatogenic cycle is shorter in chimps than in humans. The data are limited for other great apes. Third, a stronger male mutation bias would be expected in pan as opposed to homo if the ratio of male to female generation time was respectively higher. However, the opposite is true. The ratio is higher in homo than it is in pan. Phylogenetic studies produce estimates of male mutation bias that may be affected by ancient genetic polymorphism in closely related species. Even though we corrected for this effect, our results should be taken with caution because of incomplete data on the sizes of the ancestral great ape populations. Pedigree studies inferring male mutation bias are unaffected by ancient genetic polymorphism. One such study detected significantly higher male mutation biases in humans as opposed to chimps, in agreement with our results, while another study found no significant differences in male mutation biases among the great apes. These two studies analyzed only a handful of trios per species, and thus their conclusions should be reevaluated in larger studies. So this whole section, right, I've included because we're about to look at a larger study here in a moment. In the gene content evolution section, we get a few important things. One that I want to point out is across all great apes on the Y, right, which specifically is characterized here as having very low gene birth, there's only one gene that was born, like gene family that was born across all of the great ape Ys, which was VCY, acquired by the common ancestor of humans and chimps. So that simply should not be under a young Earth creationist lens. They talk about gene death rates and how they've provided some pretty solid support for like high rates of gene death in the Pannons in particular, which is certainly leading to a large amount of the disparity between the Pannon and hominin Y chromosomes, right? And they finish with their future directions where they argue that we simply need more individuals sequence so that we can know that we're looking at a decent representative for the species instead of just one individual, right? Like this was a draft of the Y for bonobos and for orangutans, but they really want to take it to the next level, which we're going to do here momentarily. That was the last paper, like chronologically, that I tended to cite on the subject, and I feel like it was pretty open and shut then, to be perfectly honest. Like here you have a massive genome, and overall humans, chimps, and bonobos share just an overwhelmingly large amount of these genomes with one another, particularly within the protein coding regions, except for that pesky Y, which we had a decent hypothesis with some decent data to support that the reason was sperm competition. But as that paper pointed out, and as papers prior to it that we've already looked at have pointed out, really what's going to sell this once and for all is like some really solid sequenced Y chromosomes from multiple individuals that are robust to get to the bottom of this. But before we get to that last paper, the one that did that really finally fleshed this all out, in my opinion, once and for all, I want to talk about a paper that came out last year in 2023 titled 80 Million Years of Rapid Evolution on the Primate Y Chromosome. Because this entire time we've been talking about like the Pannons having this sperm competition that's leading to like rapid evolution on their Y chromosome to the exclusion of the other great apes. But like that sounds just so and less contextualized with other primates to me, right? Like I want to know that that can happen elsewhere. Like it's not just being invoked for chimpanzees. And in fact, Zhao and colleagues in 2023 did exactly that. We're just going to read the abstract for this one, and then I'm going to give you a nice little anecdote to sort of round this off before we move on to our final paper. And that's also because this is not an open access article. The Y chromosome usually plays a critical role in determining male sex and comprises sequence classes that have experienced unique evolutionary trajectories. Here we generated 19 new primate sex chromosome assemblies, analyzed them with 10 existing assemblies, and report rapid evolution of the Y chromosome across primates. So this is not just a chimpanzee thing. It isn't even just a great ape thing. It's a primate thing. The pseudo-autosomal boundary has shifted at least 6 times during primate evolution, leading to the formation of a simiiform-specific evolutionary stratum and to the independent start of young strata in Catarrhini and Platyrrhini. Different primate lineages experience different rates of gene loss and structural and chromatin change on their Y chromosomes. Selection on several Y-linked genes has contributed to the evolution of male development traits across primates, male developmental traits across primates. Additionally, lineage-specific expansions of aplaconic regions have further increased the diversification of the structure and gene composition of the Y chromosome. Overall, our comprehensive analysis have broadened our knowledge of the evolution of the primate Y chromosome. When I saw this paper, I was like, oh boy, we're getting geared up for something big here, for some serious looks at the Y chromosome and specific lineages even outside of Hominoidea or Hominidae. So I emailed Zhao and I asked him, like the lead author on this paper, and I was like, hey, I want to make sure that I'm clear on this, right? This paper is suggesting that the rapid evolution of the Y is not unique to chimpanzees and that we see this in other lineages, like for instance, lineages that creationists would accept are related. And, I didn't bring up the creationist part to Zhao, but Zhao confirmed for me that yes, that is what this paper is saying. And then finally, in 2024, this year, just a couple of weeks ago, we get the complete sequence and comparative analysis of ape sex chromosomes by Makova et al. And this is the big one, folks. It's open access, so we can really sink our canines into this one. It's got an enormous author list, as you can see. We get the beginning that is much like we've seen before, talking about the nature of the sex chromosomes and how important they are. However, owing to their repetitive nature and incomplete reference assemblies, ape sex chromosomes have been challenging to study. Here, using the methodology developed for the telomere to telomere human genome, this is like, again, the human genome is 100% completely sequenced. They used that methodology for these Y chromosomes. We produced gapless assemblies of the X and Y chromosomes for 5 great apes: Panpaniscus, Pantroglodytes, gorilla, Pongo pygmaeus, Pongo abellii, and a Simang Symphalangus syndactylus, and untangled the intricacies of their evolution. Compared with the X chromosomes, the ape Y chromosome varies greatly in size and has low alignability at high levels of structural rearrangements, owing to the accumulation of lineage-specific ampliconic regions, palindromes, transposable elements, and satellites. Many Y chromosome genes expand in multi-copy families, and some evolve under purifying selection. Thus, the Y chromosome exhibits dynamic evolution, whereas the X chromosome is more stable. Mapping short-read sequencing data to these assemblies revealed diversity and selection patterns on sex chromosomes for more than 100 individual great apes. Right? Remember, that's exactly what this paper was asking for almost 5 years ago. So, good on our new guys here. We've really amped it up. These reference assemblies are expected to inform human evolution and conservation genetics of non-human apes, all of which are endangered species. The introduction tells us the usual stuff that we've already been over several times today, and they note that the human sex chromosomes have only recently been completely sequenced, and the sex chromosomes of our closest living relatives, the non-human apes, were incompletely characterized. Owing to the haploid nature and repetitive nature of the Y chromosome, most previous studies have assembled female genomes omitting the Y altogether. Ape Y chromosomes have sometimes been sequenced with targeted methods or via shotgun sequencing of male genomes, but such assemblies are usually fragmented, collapsed, or incomplete. Ape X chromosomes have been deciphered to a greater level of contiguity, for example, in different references that they cite there, but in their assemblies, particularly for long satellite arrays, they remain unfinished, preventing their complete characterization. They say previous cytogenetic studies demonstrated lineage-specific amplifications and rearrangements leading to large-scale variation among great ape Y chromosomes, for example, reference 19. The initial assemblies of the human and chimpanzee Y chromosomes revealed remarkable differences in structure and gene content, despite short divergence time, and an acceleration of substitution rates and gene loss in the Y chromosomes was observed in the common ancestor of the bonobo chimpanzee. The Y chromosome of the common ancestor of great apes probably already possessed the ampliconic sequences and multi-copy gene families in all ape sex chromosomes that share the same evolutionary strata, while experiencing lineage-specific expansion and the loss of ampliconic genes. This progress notwithstanding, the lack of a complete ape sex chromosome has prevented detailed inquiries into the evolution of ampliconic regions, palindromes, segmental duplication, structural variants, satellites, transposable elements, and gene copy number. Here, utilizing the experimental and computational methods developed for the telomere to telomere assembly of the human genome, we deciphered and completed sequences of sex chromosomes from 6 great ape species and studied their structure and evolution. So if you're a young Earth creationist right now, you should be very, very worried, because what we're about to go over completely annihilates once and for all, in my opinion, this argument for the Y chromosome being what differentiates the kinds, or being something that refutes evolution in one way or another. And they're never even that specific on it. They're mostly just like, unexpected thing, evolution destroyed. But in this paper, not only, again, are we seeing how this Y can't be used to distinguish the great apes, but also we have a mechanism experimentally shown to be viable as a reason for why these Y chromosomes are so divergent, not only within the great apes, but as we saw in the previous paper, probably at play within the order of primates as a whole. So this figure 1 is the crown jewel, right? We have over here on the far left a phylogeny, and then figure 1b, we have a heat map for pairwise comparisons on alignability, not sequence identity, but alignability of the X chromosome here and the Y chromosome. As expected, the percent that's alignable here on the X matches pretty much with the rest of the nuclear genome. You're looking at the bottom organism compared to, like, the organism on the X-axis here compared to the one on the Y. So for instance, the percent of the human that can align to the chimpanzee is, as highlighted by my cursor here, 97.6%. on the X chromosome. The percent of the Simang Y that can align to the human Y here is 84.1% on the X chromosome. It's pretty conserved, which is why most of these are pretty similar. And everybody is going to be roughly as similar to the Simang as everybody else as far as the great apes go versus just a regular lesser ape here in the Simang. So creationists argued for a long time that the Y chromosome was what separated the humankind from the ape kind. So you had the human and then you had the ape with regard to this Y chromosome, that being maybe one of the separating factors. This figure blows that idea out of the water, right? Because all of the great ape Ys are extremely different from one another, directly in line with the evolutionary prediction. And this is particularly the case with any given great ape compared to the bonobo and chimpanzee, the panins, who are the ones who are undergoing that rapid evolution due to sperm competition. So here's the human right down here. The human alignability to the chimpanzee and the bonobo is like roughly 26%. The human alignability to the gorilla, less than that at 25%, followed by the Bornean orangutan, the Sumatran orangutan, and lastly, the Simang. The gorilla's alignability is significantly lower than the human, again, in line with evolutionary predictions, with the humans being the sister group to the panins, the hominids being the sister group to the panins. The gorilla alignability to the bonobo and chimp is like roughly 20%, followed by 23% to the human, 19% for the Bornean and Sumatran orangutan approximately, and 12%, even lower, for the Simang. I'm going to make this as clear as I possibly can be. If you want to say the fact that only a third of the human Y chromosome can align to the chimpanzee means that humans and chimps can't be a part of the same kind, then gorillas and chimpanzees can't be a part of the same kind. And Bornean and Sumatran orangutans and chimpanzees, the panins and bonobos, cannot be a part of the same kind, right? You're up a creek on this one. The Y chromosome cannot distinguish between a human kind and an ape kind. That argument is dead. But let's do a little compare and contrast game here, right? Because evolutionary theory made a very specific prediction several papers ago, which is that if sperm competition is to blame for why panins are so divergent from the rest of the apes, and specifically the hominins, then what we should expect is that drive, that pressure should continue to act within the panins, and it should lead to them being much more divergent than other pairs of closely related apes. And we have a wonderful example of that on display here, an evolutionary prediction continuing to be supported by this data. Because when we look at the chimpanzee and the bonobo here, the chimpanzee is 65.3% alignable to the bonobo. These are species that are as similar to one another as we are to Neanderthals. That's insane. And slightly less than that when it comes to the bonobo being aligned to the chimpanzee at 59.6%. These guys are so similar to one another in the nuclear genome, and yet their Ys are incredibly divergent. Okay, that is directly in line with this idea that sperm competition is driving it, that sexual selection within the panins is causing their Y chromosomes to be further diverged from one another and further diverged from the rest of the apes. But we have a great comparison here because Bornean and Sumatran orangutans are like roughly the same distance from one another as bonobos and chimps are. And when we look at them, presumably not with sperm competition at play, no one has proposed that for these guys, they are more similar to one another. Now, all of the apes have been characterized by rapid evolution on the Y, as we saw with the paper on the primates generally. It's just even more extreme in chimps and bonobos. So while they are pretty divergent, they are less divergent than they should be if sperm competition is at play. And that makes sense because sperm competition is not as at play in these orangutans as it is for the chimps and for the bonobos. There is so much good stuff in this paper, so let's get to some of it. In the section titled High Interspecific Variation on the Y chromosome, we get across all pairwise species comparison, the percentage of sequence aligned was lower for the Y chromosome than for the X chromosome. Only 14 to 27% of the human Y chromosome was covered by alignment to other ape Y chromosomes, whereas as much as 93 to 98% of the human X chromosome was covered by alignment to other ape chromosomes. The same pattern was observed for closely related species, with only 60 to 87% of the Y chromosome, but more than 95% 25% for the X chromosome aligned between them. So they're basically saying even with the Borneo and Sumatran orangutan or bonobo and chimpanzee, that pattern holds. It holds across all of the apes, that the Ys are less similar because they're rapidly evolving in all of the apes, and even more so within the Pannin. So they talk about some of the specific sequences here, and then they get down to this, because this is really cool. They say, in the sequences with interspecies variation, 83 to 86% of base pairs on the X chromosome, and here, this is insane, 99% of bases on the Y chromosome were affected by large-scale structural variants, and the remaining base pairs, that's 1% for the Y chromosome, were affected by single nucleotide variants. Inversions were abundant on the Y chromosome, consistent with palindromic architecture. Inversions and insertions were approximately 8-fold and 3-fold longer on the Y chromosome than on the X. So we're getting all sorts of just insane rearrangement going on the Y. But despite all that, further down in this section, it says, the phylogenetic analysis of multiple species alignments for the X chromosome and separately for the Y chromosome revealed the expected species topology. That should not be the case under young Earth creationism, you guys. This means despite the vast divergence and differences on the Y chromosome, it still spits out the same evolutionary phylogeny that the X spits out, that the entire rest of the genome spits out. But it gets even crazier because this paper is confirming what that 2020 paper also proposed, but they detected higher substitution rates on the Y chromosome than on the X chromosome for all the branches consistent with male mutation bias. For instance, the human chimpanzee divergence was 2.68% on the Y chromosome and 0.97% on the X chromosome. For the Y chromosome, we detected an 11% acceleration of substitution rates in the Pan lineage and a 9.2% slowdown in the Pongo lineage compared with the substitution rates in the human lineage. And for the X chromosome, substitution rates were more similar in magnitude among the branches. These results indicate a strong male mutation bias for Pan and a weaker bias for Pongo than for the human. Strong male mutation bias in the Pan lineage is consistent with increased sperm production due to sperm competition. Baby, this is science at work. Now, friend of the channel and population geneticist Zach Hancock would want me to note a few things for you here. The paper proposes that most of the evolution of the Y as far as architecture goes, and specifically with regard to the ampliconic regions, is due probably to purifying selection. That doesn't mean that sperm competition isn't also at play, it just means it's probably polygenic. So at the end of the day, what we have here is a fulfilled prediction by evolutionary theory that explains a discrepancy that previously didn't have an answer. Contrary to that clip that I showed you earlier, the disparity in the Y chromosomes for humans versus chimps was never preclusionary data. It was an unanswered question. Preclusionary data is when you have data that makes something else impossible. There was nothing ever presented about that disparity that made a common ancestry between humans and chimps or all of the apes impossible. It was just a big ol' huh? And yet we did find the answer to the question, and we did it using evolutionary hypotheses, predictions, and methods. So like I said earlier, there is no way to use the Y chromosome to separate humans as a kind from other apes together as a kind. Now I know what you're thinking. You're thinking, well, why don't we just have 4 kinds within Hominidae? The human kind, the Pannins, the Gorillans, and the Pongians. And to that I say, no, that's not going to work either. If you will recall, the purpose of created kinds in the first place was to sort of solve a problem that these guys had with regard to a 6,000-year-old Earth and modern biodiversity. They need kinds to be broad in the first sense because they don't want to have to take too many kinds onto Noah's Ark, which that's what the Bible says, that Noah took 2 of every kind of animal. So considering you have to have all extinct and extant animals like accounted for if they have the nefesh, the breath of life, you want these categories of kinds to be broad so that Noah doesn't have to take like one of every species of dinosaur. He can only have to take one of every family of dinosaur, every family of mammal, or every family of temnospondyl, or whatever you want to go with. But then the kinds also can't be too broad because after Noah's Ark lands, they have to account for all of the biodiversity that we see in this post-flood rock. And the broader the kinds, the more hyperevolution has to happen in just 4,400 years. So you see, they're stuck between a rock and a hard place here. What that means is that if you want to look at genetics and say, all right, all of these apes are really, really close to one another, like they're all over 95% similar, which, need I remind you, is more similar than African and Asian elephants are to one another, lions are to tigers, and rats are to mice, all of those pairs of which creationists insists are the same kind. So if you're going to separate these guys out genetically and say that despite the fact that all the apes are extremely genomically similar, if you want to say we've got Homo sapiens as a kind and the rest of the hominins, panins, gorillans, and pongins, well, that becomes the standard. And you then have to parse out every other family level kind using that criteria. A criteria which effectively becomes the genus level. Do you know how many genera of animals there are on the planet today? Well, according to Biology Stack Exchange, there are 73,540 genera just alive today, and they have to account for 550 million years worth of genera. Well, now that's just a little bit different than the few hundred created kinds currently accepted at Answers in Genesis and other organizations. Anyways, my gentle and of course very modern apes, this has been a fun one for me. It's always good when I try to brush up on my genetics. There may be mistakes in this video. I hope that's not the case. I'm gonna try to run it by some folks first. The next time you see someone try to use the Y chromosome as an argument against common ancestry within Hominidae, what I think you should do is point out the fact that Y chromosomal evolution is extremely well characterized within the order, and that if you want to use that as a means by which to debunk evolution, one, you're going to have to do something about all of those accurate predictions associated with this line of inquiry, and two, you're going to have to either accept humans as members of the ape kind with the rest of the hominids using the Y chromosome, or separate the apes out into so many different kinds that when applied broadly in a standardized fashion across the animal kingdom, you break young Earth creationism. I think the most deadly part of all of this is that even using the Y chromosome, you still end up with the conventional phylogeny, and also the fact that humans are still more similar to chimpanzees. It's just gorillas are more similar to humans than they are to chimps. If you liked this and if you like what I do generally, please consider liking, commenting, and subscribing. That helps the channel in a major way, and you know, I love helping the channel out. I don't do sponsorships or anything like that because I want this channel to be a fun and enjoyable place to learn. So if you feel that that is a worthy cost to support, or just like the type of content, supporting me in the free way is always appreciated. And if you want to support me in a more like financial way, you can do that too by becoming a member of my Patreon, which like patrons get early access to videos some of the time, and they also get their names at the end of every video. So both of those are kind of cool perks, I would say. And I think I'll just close with this from the bottom of my gentle and of course very modern heart. I want to thank you guys for being curious primates, and I'll see you really soon next time.